Assalamualaikum warahmatullahi wabarakatuh. Welcome everyone. My name is Lutfia Hastiani Muharam. I'm from Biotechnology Department of Universitas Muhammadiyah Bandung. In this virtual moment, I'll present our research as a participant on international seminar Paris Panjapa that held by Universitas Muhammadiyah Tasikmalaya. Okay. First, I say thanks to Allah that give us joy of Iman, Islam, and health, and also salawat and salam for our Prophet Nabi Muhammad Rasulullah Sallallahu Alaihi Wasallam. Okay, here our research presentation. Okay, the time of the research is isolation bacteria producing alpha-amylase from Black Soldier Fly Larvae, Hermetia ilucens. The member research were me, Lutfi Hasyani Muharam, my partner Elis Hernahadini, and Muhammad Fauzi. We are from Department of Biotechnology, Universitas Muhammadiyah Bandung. Okay, this research were corporate program besides Universitas Muhammadiyah Bandung and Cimahi City Government. Okay, this is the content presentation, and here the abstract. Okay, today Black Soldier Fly Larvae, well known, most effective for organic waste bioconversion. The ability BSF larva to reduce organic waste is up to 200 tons per day. Oh, what's a numerous number, isn't it? Maybe, I'm sorry, why they can do it? Okay, because the larvae contain many microorganisms, and the microbes in the larva gut have so many functions that are crucial for larva development. Okay, this is the life cycle of BSF larva. Okay, we know that the longest time life is a larva stage with lifespan 13 to 18 days and consists of 6 instars, and then the larva become the pupa and then adult insect. Okay, next, alpha-amylase. Alpha-amylase is one of the important enzyme in development of biotechnology and belongs to the main enzyme class on the world enzyme market. Alpha-amylase from microorganism was more perspective than plant and animal amylase because it was more easily manipulated to produce targeted enzyme characteristic and more economical. Okay, that is the reason why we prospect amylase from microbe BSF larva. And this is the example of three-dimensional structure of microbial amylase from Anoxibacillus. Okay, next about alpha-amylase mechanism. The amylase enzyme will hydrolyze glycosidic bond of polysaccharides from starch, becoming simple sugars, glucose or maltose maybe. Okay, here the experimental. This research was conducted in microbiology laboratory Universitas Muhammadiyah Bandung and Research Center of Bioinformatics and Biotechnology Universitas Pajajaran. And the sample we get from organic waste bioconversion by BSF from Padasuka Village, Cimahi City, West Java Province. Okay, this is the step of experimental, and this is the detail of procedure. and results and discussion. Yes, sorry. This is in our research, we investigated the part of larva body and tests the activity of amylase. Okay, this is the fresh larva, and we dissect them, and we got three component of body of larva BSF. The first is cells, the coating for body. The second is body content rich of lipid, and the longest one is that rich of protein. And alpha-amylase assay, we assay alpha-amylase from crude extract and obtain the higher activity of amylase is from body extract than gut extract. Okay, this is why as our body, the biochemical process of basal larva starts from mouth, and in the mouth the salivary glands secretes the enzymes amylase and maltase. Besides that, food decomposition occurs too in the middle of the stomach. Now, this is the detail of schematic summary of important processes in BSF larva. The first scheme is larva reactor. The larva reactor is in the gut. Okay, and the second scheme is gut compartment. There are three components of gut: anterior, mid, and posterior, and the mid gut is compartmentally longitudinally considering pH and enzyme. And there are so many reaction of enzyme and microbes decomposition. This is the mechanism of microbe digest or by convert the macromolecule to simple molecule. Okay, so the microbes such as fungi and bacteria produce some enzymes and the other metabolites, and the enzyme and metabolites will hydrolyze the macromolecules to simple molecules such as fatty acid, amino acid, and so on. Okay, next. This step was proved the theory from slide before. Then we got isolated bacteria from body extract of BSF larvae. There are obtained 8 different single colonies based on morphology of each colony. And the 8 colonies we ground in second incubator to produce the enzymes, and we harvested by centrifugation to obtain the crude enzyme extracts. We know alpha-amylase is an extracellular enzyme, so the enzyme obtained at the supernatant. Okay, the enzyme extract then investigated of alpha-amylase activity by flow method and gave results as in figure 9. Okay, this is the results. This is results of amylase activity unit by FUA method. This is the first data. The highest is from crude enzyme. So the all of bacteria contained here. So the result is very high. Okay, and the remain is where the is 8 of isolated colonies, and the highest activity is 8 colonies, 8 colony or M8 colony. The activity is 10.9 unit per milliliter. Okay, this is the reason about our discussion about the results. The various of activity amylase shown that is isolated bacteria from BSFL have different characteristics. Okay, this is can be different characteristic. It can be used as preliminary research to explore bacteria biodiversity from BSF larvae. Previous research review gut bacteria dominated by Pila vermicules, proteobacteria, bacteroides, and actinobacteria. Okay, and the level of amylase production varies depending on genus, species, and strain. Besides that, the source of the microbes' origins and environment factors such as pH and temperature can affect the production of amylase. Microbes that isolated from starch or amylase-rich environments naturally have higher amount of enzyme, of course. Based on sorts of BSF larva samples, our samples from Padasuka village, Cimahi City, the feed of BSF larva was organic waste from hometown behind among the area Padasuka. So the dominates by food scrap such as rice, fruits, and vegetable residue. This is the conclusion. Microbial enzyme is significant research field to explore biodiscovery and bioprospecting enzyme for industrial application. The present study clearly illustrated that Black Soldier Fly Larvae have a great ability bioconversion, and it's associated with microbial diversity, primarily in the gut. This study have been isolated bacteria colonies from BSF larvae and proved the value of alpha-amylase activity. The highest activity level of alpha-amylase was obtained by 8 isolated bacteria, which the value was 10.9 unit per milliliter by FUA method. These isolated bacteria can be further researched to identify genus and species, and also the most important is to genetic engineering studies to get optimal perform enzyme for industrial requirement. Here are the references. Okay, acknowledgement for Universitas Muhammadiyah Bandung, of course for Cimahi City Government, Faculty of Science and Technology, Department of Biotechnology. Big thanks to our team. We named Cimahi Zero Waste Team. Sorry. The team for Mrs. Yayulfa, Mrs. Korea Tursuhada, Mrs. Nelis Hernadini, Mr. Muhammad Fauzi, Mr. Algi Faruni, Bapak Mustafa, and me, Lutfiah Stiani Muharram. And big thanks to biotechnology students for the helping working laboratory. Okay, and thanks to for organic waste BSF conversion area at Padasuka Village, Cimahi City. By this experience, we can conclusion that the larva BSF is the solution from Allah Subhanahu wa Ta'ala to reduce the organic waste. Okay, and the larva can make the zero to hero, from the dirty to green. Why? Because the larva BSF is source of protein, the most protein source for feed of cattle, fish, and the residue of composting is very the best for agriculture. Okay, so the larva BSF is very interesting to development. And thank you for your attention, and for the Committee of Paris Java International Seminar. See you the next moment. Assalamualaikum warahmatullahi wabarakatuh.